Should I just start, or you want to talk?
No, I'll give you a little.
Oh, okay. Oh, right, right. It's pretty there.
Yeah, no, I didn't know where I was going to go. I didn't know where I was going to go.
Well, I think there's more dams and covers. That's not where we go.
I don't even try to
One on Main
Wireless fails, computer dies, electricity goes out. I still have
Yeah, that's where the Y is. Okay.
I mean, that's what it's gonna be.
Yeah.
Like,
I used to go there quite a bit, because they were open a lot. Oh, yeah. Oh, yeah. That's amazing, because
Did you notice last night that my devil boss had sheared?
I tell them the same
No.
thing.
We got a good
I
rip.
don't take that anymore.
Right now I'm
And
going,
it just keeps coming up.
there's a
I
show
said, no,
here
I
on
haven't
the
taken this
corner
for
on the
years.
Oh, no.
So they eliminated.
But I haven't
it.
really, I lean, that's leaning hard, I don't think, it's probably just cracked. Yeah. I think I can make the fall fixed, but.
Yeah. Yeah. Oh, okay. You probably can't remove it,
Well, yeah, you
You
can.
just got to
But
look
it
at
just-
the special one.
but it still doesn't. Right. Right. So I don't know what they're. Yeah. Right, right.
Next day it comes in, we are in process on this prescription.
Oh, yeah.
I managed to go golfing 4 times this year.
This day it's already ready.
Yeah.
19-1, the heavens declare the glory of God.
Yeah.
I was guessing the songs, but I wasn't sure.
Right.
Somebody that.
You know, it's kind of like, we buy another van, it might be a year old, so it's not brand new. And a week later, we get a note, would you like to buy another van? Yeah, like, just buy a van every week, huh?
It rises from one end of the hill and circles to the other end.
Trade up.
Yeah, right. You didn't notice that? You don't see all the time, huh? Yes. Who did you buy yours from?
I think it probably.
The Subaru, it used to be Cliff Raw, there on Mason.
Good morning.
Good morning.
I am, it was great. We had a really good time.
I forgot where you went. Wow. The family? For all our oil changes. Big. And this Tom's Tom. They were.
Is it recorded?
I don't know.
Listen, Maxwell.
Oh, this is gonna be good.
Now they got this thing that started at 12.
Drinking water out of those fire things.
Okay. Watch it again.
I don't know why.
You gotta register.
I really have these talks on the ground.
What's that?
We have these talks on the ground
Okay.
because we've given them a Zoom call.
Yeah.
This one, not Tom's.
So everybody registers.
Some of the other ones we've given them.
Every year.
I can send you
but it's just my name, Jennifer Levin, L-E-V-I-N,
Okay.
at Gmail.
Good morning.
Because I can't remember the name.
Sure.
It should be on there. Okay. Yeah.
And I have a paper on there with questions.
Sure. Oh, okay.
Register cat.
The keys,
Yeah, I'll look at
the keys on the screen.
Oh, great, okay, perfect.
That way I wouldn't lose it.
Well, I'm gonna leave it there for a second. I have the other set of keys. Okay, I will look that up. Thank you very much. I know it was in good hands.
Good morning.
It's happening.
It's early, but I don't know how many next week can have to do this. And it's too big, I didn't know. So you can open
I think if you do it regularly, you realize it's easier, and then you kind of surround yourself. I mean, the bank is not going to foil.
Right.
I mean, this is the third time it's been done. So I have had where they said chocolate overdose, chocolate overdose. That is, yeah, chocolate.
But you'll have to laugh at the mistake that I made on this big
I've done that before. I just, at the time, I would just email Charlie and be like, well, then, that I crossed that out. I made a mistake, let's cross that again. As long as it's on there and,
Yeah.
all, I've got enough.
How was your week? It's good. Your vacation?
It was
We had all our
we had
family
a
there.
wonderful
You know,
time.
the
My great
Peter and
uncle
the kids.
went
Yeah,
from not really doing so good.
Yeah, Sue came up, and her sister came up for a few days, and then they all left yesterday. So I've got, I washed all the, actually walking around. I got cheese for bunches of beds. It's amazing. You know, that is Mars.
It does, and I think you kind of try a little more, not that you're not trying.
Yeah.
But
And I would have a lot of people.
No other name is power to save, but Jesus Christ the Lord.
That's right. Yeah. 13 hours away, is rather have them come here.
As I contemplate your suffering on the cross and rest ahead, the fury of the Father's wrath poured upon your head. My mind cries out in unbelief that you endured so.
All right, good morning. Welcome to our Discipleship Hour here at Lakeshore Baptist. It's so good to be with you. We had a good time away a little bit this week, and we're glad to be back. We've got something special planned for you today. Many of you know Tom and Betsy have been involved in the intelligent design movement in different ways, but God has just given them an ability and an understanding in ways that I think is beyond some of us, including myself, in some of these issues. So I have asked, you got the chance to hear Tom a couple of weeks ago, I've asked Betsy to come this morning and give us a presentation, and you see what it's called here. It's a tour of a tiny city, and this is about the cell. And what Tom did a couple of weeks ago is he kind of took a big look, right, at like this wide view of the universe, and how you look at this big picture of how, there's just us. And the fact that it's just us in this massive universe is an evidence of God's creative work and how that all kind of leads us. So where we got out the telescope before, now we're going to get out the microscope, and we're going to look, instead of the big look, we're going to look like down to the stuff you can't even see with the human eye. And everything, it's interesting how everything from from the biggest expanse of the universe down to the tiniest part of the cell, everything declares the glory of God. And that's what Betsy is going to do. So I've asked her to come and do this for us for Discipleship Hour, so you guys can get ready. And she's got some great info for us that'll help us understand some of God's creative power and wisdom. So Betsy, why don't you come?
Yeah, I was going to use that same verse about the heavens declare the handiwork of God. And again, this is what I'm going to be showing you here. They wouldn't have known back then because they didn't even know what cells looked like at the time. So I'll start with a conclusion for this talk that the cell is very complicated, and it could not have evolved once you see. And I'm going to be throwing a lot of things at you that you're just get the big overview, because there's going to be a lot of things that are technical, and just get us, and come ask me afterwards if you don't understand what I'm saying. So anyways, a tour of a tiny city. So cells are the building blocks of all organisms. Here's my pointer here. Handy dandy yardstick. So in single-celled organisms, there's like the yeast, the amoeba, some algae, and then there's the multicelled organisms. Pretty much everything you can see, touch, whatever, are all multicelled organisms. And then the slime molds are a real interesting organism. What they do is they are single-celled organisms unless they get into starvation mode. And then when they're in starvation mode, they go together, build a multicelled organism with reproductive organs, muscle organs to be able to move, cilia to move and things. And then once they're got food again, then they break up and be single-celled organisms. So they're pretty interesting kind of a organism there. So they didn't know much about the cells way back when. The first one was Robert Hooke just coined the term cell because it reminded him of the monks lived in monasteries, and each one had their own little room with their own little bed, and it wasn't any big rooms, and each one of those little rooms was called a cell. So when Hooke looked in the microscope, and here's a picture of what he probably saw, he called them cells because it looked like the place of the rooms that the monks lived in. And then in 1833, so that's almost 200 years later, they were able to see the nucleus in the cells. But that was like 200 years later. And then in 1835, just shortly after that, they saw, here's a cell, the nucleus, and then this dark spot. Am I standing in your way? They saw the nucleolus. That's all they knew about the cell. So when Darwin came along, that's all he knew about the cell. He knew that he saw that it was a bag of protoplasm. He knew about the nucleus, and he knew about the nucleolus. That's all he knew. So when he was talking about, he could see in the sky the evidences of God, but in the cell, the biology was not there for him to understand what was so complicated about it. They also had the idea, they didn't know about genes, they didn't know how inheritance worked. They thought at that point that it was called acquired inheritance. That, for example, if a giraffe had to stretch her neck to get to the higher leaves, if she was carrying a baby, then that baby would be born with a longer neck because the mother had stretched her neck. And so they didn't know that genes existed or there was any kind of discrete thing. They thought the inheritance was something continuous and easy to acquire. So that's what he knew. That's what Darwin did not know. And we're going to go through a lot of those pieces, not all of them, but I'm going to go through some of them. So, what does a city need? I want some input from you guys, what you think when you think of Kewanee. What kinds of services, what kinds of places that you would need to make a city? Any ideas?
Power center?
Power center, yep.
Grocery store? I could, yeah, grocery store. Water. Roads. Good. So, there's the power center, power plant, streets and roads. And the whole cell is filled with water, so it already has the water, so we don't need a water plant. But this is City Hall, library, moving company, manufacturing plants, post office, waste treatment plant, power plants, storage facilities, streets and roads, city gates. And in addition, this city has the ability to reproduce and to move around.
Does it also have a protective system?
Oh, yes. Yes, this is some of the stuff
Yes.
that we're going to cover. So we'll start with the City Hall, which is the nucleus. The nucleus contains all the information, almost all the information you need in the terms of DNA. It's stored in the information stored in DNA. So all of this section here is the nucleus with DNA in it. Here's the nucleolus, which is the center. I'll get to that one in a little bit. And there's nuclear pores, these things that just look like little holes. They're not really little holes. They're another organelle in there. And this is a highly guarded, things don't go in and out of the nucleus without going through the nuclear pore, which is quite complicated just on its own. In fact, the lab I worked in, one of the proteins we worked on, one of the genes we worked on was one protein in the nuclear pore. And if that one protein wasn't functioning properly, the whole cell would shut down. So that's how important a nuclear pore, one protein in a nuclear pore is. So, nucleus is also a library. It stores information. Like I said, all the information, almost all the information is stored in DNA. And then it goes from, here's inside the nucleus, and it goes from DNA, it gets transcribed into mRNA. Now there's different kinds of RNA. This is an mRNA for messenger RNA. And so then it takes it from DNA to mRNA, goes out of the nucleus into the cytoplasm, and that gets translated into protein. So we'll look at that process. So if you're in the library and you want to find some information, you ask a librarian. So the librarian I'm calling is a transcription factor here, and we'll go into more detail there. So here's the gene, start of the gene, goes down this direction here. And here's a transcription factor binding site, and the transcription factor is another protein, comes down, sits on that, and we'll look into that piece. Let me see if I'm missing everything. Oh, the other thing is, so if you think of all the cells, you've got liver cells, you've got skin cells, you've got muscle cells. All of them have the very same set of genes in them. How does a liver cell know to make the liver cell proteins, and the muscle cell knows to make the muscle cell proteins, but not make liver cell proteins if you're in a muscle cell? And that's done by the transcription factors. So transcription factor is a protein, like I said. So the gene starts downstream here. This is upstream. And the transcription factor binding site is a specific sequence of 6 to 8 bases that will be recognized by the transcription factor. And for example, if a cell goes into heat shock, that it gets, the temperature rises, and that will destroy the cell. That instigates a lot of heat shock proteins that get translated. And those are released because all the transcription factors that are needing to make the proteins for the heat shock proteins get produced, and then they will sit down on this transcription factor binding site right here. Once that gets set down there, it recruits about 6 to 8 other proteins that come in because of the transcription factor, And this becomes the polymerase that goes downstream and translates the DNA into the mRNA. And then when a gene is transcribed, again, we'll go look up here, the DNA is not contiguous for the recipe for the protein. There have exons, which are the patterns for what you need for the protein, but in between there's introns. Those are controlling factors, they think. They're not quite sure what all of the introns do. But you have to cut them out so you can have a contiguous mRNA. And so this gets translated into this transcript, but then it has to be, these introns have to be removed, and that's done by something called the spliceosome, another little micromachine organelle. And what that does is there's a 2-base signal here and a 2-base signal here, and the spliceosome cuts the DNA here, cuts the DNA here, or the mRNA here, puts it back together, and the intron gets thrown out. And then that gets translated into the mRNA, which goes out of the cell. Here's a nuclear pore. There's an mRNA shooting out of the cell, and it goes out pretty fast. You can just shoots out. And feel free to ask any questions if you want. So how do we get from RNA to proteins? So we're now going to be looking at this part. Now we're in the cytoplasm, and we're going to look at translating from mRNA to an actual protein. And that's done by the ribosomes. And the ribosomes, I think there are two, maybe the most complicated organelles in the cell. One of them is the ribosome. And the ribosomes are all these little tiny dots here. They're filled, this whole thing is filled with ribosomes because they're busy making proteins from the mRNA. They're producing that. So here's kind of an artist's rendition of what a ribosome looks like. It's, they've been able to see it because they have techniques in electron microscope. The electron microscope is even, they have high pressure, freeze drying, fast. The EM micrographs really have gotten really good, so you can actually see some of these tiny little organelles. So that's what it looks like. Here's the mRNA. This ribosome is reading the mRNA and producing the protein out here. So here's the cartoon that we'll look at more carefully. So this is done very quickly. It's about 20 amino acids a second that they can put, create the amino acid. So here is the small subunit of the ribosome, the large subunit, and it has 3 sections here that are kind of the manufacturing part of the ribosome that it translates the mRNA into the protein, 1 amino acid at a time. These little things that look like pawns in a chess set, this is the tRNA, which looks more like this. This is another type of RNA. The T looks, because this looks like a T, but also a T stands for transfer. So what this tRNA is doing is, so the RNA and DNA are made up of nucleic acids. That's a type of a protein. But then it needs to be, well, not a protein, a molecule. But it has to translate from bases to amino acids, which is a different type of a product. And so this is translating the RNA into protein by going through this cartoon here. So here's the mRNA going through,
and it will read the amino acid is on the top of the tRNA, and it comes down in here, and if these 3 bases match up with the 3 bases down here, it hooks up and moves over to the second section here. If it doesn't match up, it gets kicked out, and another one comes in, 20 amino acids at a time, per second. So it comes down here, and if it reads it, it bonds to this, gets moved over here to the second section, then this is the amino acid gets pushed onto here and formed, and then when that's done, then it goes here and gets kicked out. And meantime, the next one comes in, and it's a very, very fast process. So how are the ribosomes made? So the ribosomes are made up of ribosomal proteins, and here's another kind of RNA, ribosomal RNA. And so then in the nucleus, right here, this piece, there's DNA that translates into messenger RNA, including the mRNAs that code for the proteins in ribosomes. And the DNA in here also codes for the transfer RNAs. In the nucleolus, the DNA codes for the ribosomal. So that's a function of the nucleolus, is that's where the information is for the ribosomal RNA. But the real question is, what's really crazy is ribosomal proteins are made by the ribosomes. How could that have happened by chance? It's, the thing you're making is what you need to make it. So what happens when a protein is made? How does it know where to go? Mr. Osborne, may I be excused? My brain is full. So there's post office. The endoplasmic reticulum and the Golgi apparatus are different other organelles that code and put post office tags on these proteins to tell them where to go. Once they're made, they have to go someplace in the cell or out of the cell or what, they don't know what, so they're tagged. They're the endoplasmic reticulum. Here's the Golgi apparatus here. There's smooth endoplasmic, I'm going to just call it ER, smooth ER, and rough ER. This is called rough ER because it's just got the ribosomes attached to the endoplasmic reticulum, and here the smooth ER does not, but they're pretty much the same thing. And what the endoplasmic reticulum does is it tags for transport, metabolism, where the protein should go. Maybe they go for metabolism or steroids. And then there are some proteins that are tagged to go out of the cell, like antibodies. If you've got a cell that's making antibodies and it has to get out into the bloodstream, then those are set for going out of the cell. So here is endoplasmic reticulum, and then it goes, if it's set to go out of the cell, it goes through the Golgi apparatus, which encodes it into a little cell or skin around there, and then it gets into a secretory vesicle, and then it gets sent out of the cell for, the antibodies or whatever. So where are we so far? We saw the City Hall, library, manufacturing plant, post office, and now we're going to be looking at the streets and roads, moving company, and the storage facilities. So these are made up of microtubules, the roads are. There's mainly two types of tubulin. Microtubules are made up of tubulin, and there's an alpha tubulin and a beta tubulin. And they'll fit together 13 of these tubulins, every alpha, beta, and it's kind of this spiral, and it makes these long, skinny tubes, which are kind of ropes in a way, but they are made 100 monomers per second, but they can depolymerize at 1,000 monomers per second.
And one of the functions of the microtubules is it provides structure for the cell. One of the other uses is that acts as a street or a road. This guy here is a protein, kinesin, and it really does walk along the microtubule. And here's the storage facility. So here, this is full of whatever's going to be secreted outside the cell, either antibodies or some proteins or whatever. And so this thing walks along the cell. It really does. I've seen, they take pictures of this protein in microscopic, micrographs, and then they put them together, and this thing just walks like this, carrying this other guy. And here's another one in the background here. And then this is a storage facility, so that's another function of the city. And then a moving company. So I got to explain this picture. This is a picture of what's inside the nucleus at the time of cell division. So when a cell it starts to make two cells, first it has to replicate all of its DNA. And then all the DNA lines up, the types of DNA, one all the way down, and that's this blue here, is all the chromosomes lined up right there. These are the microtubule organizing centers. They've sent out all the microtubules here. And what happens is the microtubules will grab onto a piece of the chromosome and in a certain spot there, and then it starts to depolymerize at this point, but it's grabbing onto that point. It doesn't let go. And so as this depolymerizes, these things get shorter and shorter. It pulls the DNA apart. Once the DNA is apart, the cell wall comes down through here, and you've got a new cell with DNA in each of the two cells. So that's the moving company function of the microtubules. We're going to be I'm going through too fast. We're going to have a lot of time left over. So last three we'll cover. The waste treatment plant. So this is the lysosomes right there. And the lysosomes are very acidic. If there's pieces of things that were made but not made correctly, they will be put into the lysosome, gets kind of digested, and then sent outside so that it's not just cluttering up the cell. And they'll break down excess worn out parts, or if they're not made right. There's also, if something's going wrong with the cell, there's another system that's set up that it will self-destruct. The cell will commit suicide. If something's drastically wrong, it just stops, and all these other proteins are started to self-destruct the whole cell. And that's called apoptosis. What happens, that's one of the problems when you have cancer, is that loses its ability to check if the cell is working properly. And in cancer, quite often, that proteins, there's a P53, P21 are two proteins that monitor the cell and say, is it right, is it wrong? If it's getting the wrong number of chromosomes aren't working right, there are too many in this side, they're not splitting properly. These two proteins, when they're working properly, will stop the whole system down and start sending out the proteins to say, time to commit suicide. And that whole system works. In cancer, if that genes aren't working right, that's when you get cancer. And I had a young friend of one of our kids. She was born with the P21, having a defect in P21. And I think she got her first cancer when she was 3, and by the time she died, I think she was like 27, she had about 5, 6 cancers because she was born without one of these monitoring genes. So they're pretty important. So the other most complex organelle is the mitochondria, which acts as a power plant. So we get all of our energy from the sun via plants. So a plant will take the water from the soil, carbon dioxide from the air, and sunlight, and transform it into glucose, which sometimes gets translated into starch or other things, and oxygen, which the oxygen goes back into the air. So they take the carbon dioxide, the water, and put oxygen out. And then when we take the glucose in this version of either pasta or the meat, because the animals have eaten the food that has the glucose in it, so we get the glucose eventually. We get energy out of the glucose plus the 6 oxygens of molecules of oxygen, and we produce energy, our cells do energy, and then water and carbon dioxide. So you've got this complete cycle here. There's a little bit of loss, but not a lot. It's just a complete cycle. You start with water, carbon dioxide, and energy, and you end up with energy, water, and carbon dioxide. So we'll look at the function of that, how that goes. So here's the mitochondria. Again, this is a little organelle in the cell, and there's quite a few of them in there. And here again, you're in the cytoplasm. The mitochondria does most of the work, but before it gets to inside the mitochondria, it does most of the work, some of the work outside in a glycolysis. It's called the process here. It's called glycolysis, and it takes one molecule of glucose and ends up with 2 molecules of something called adenosine triphosphate, which is ATP, shortened for ATP. The energy that it takes out of the glucose molecule, it transforms that energy into ATP, and the bonding in the ATP is what our cells can use. We can't use the bonds in the glucose directly. It has to be transformed into the ATP, the bonding in the ATP. So this one produces 2 ATP. When we were, this past winter, we went down to a conference in Texas on intelligent design. The gal who presented this, spoke an hour on just this process. Because it's, again, all of this is really, really complicated. It's probably giving you about 2 years worth of molecular biology information here. So then it goes inside the cell. And again, this is a very complicated, it goes around, it's called the Krebs cycle, and it'll keep, I think it goes around 3 times, and it gets all the rest of the energy from the glucose bonding and sticks it into the bonding in the ATP, which our bodies can use. Again, we can't use glucose directly. And so this has a lot of different proteins coming in, proteins leaving, all sorts of things to get the ATP out. And here's an ATP synthase. This is a membrane within the mitochondria. And here's a blown up picture of a little molecular machine inside of the mitochondria. And this rocks back and forth. It gets pushed back and forth here. And that changes what's going on here. And that provides the energy to put the ATP, the energy into the ATP molecule to bond it. I don't know how it works, but I thought that was a cool picture. And it just, again, these are all machines that Darwin had no idea about. Most of this stuff was learned, probably the electron microscope started, like they got it around the 1950s, 60s. I started working in the early 70s in the biology labs. And at that time, they were just discovering how to manipulate genes. They could sequence genes, but at that time, somebody going on for a PhD in biology, they spent their whole thesis on sequencing one gene. Now you can take the gene, take it out, send it off, get it back in a few days, and you know the sequence on it. So in, what is that, 40 years? 30, 50 years? 50 years. Okay, 50 years ago, most of this stuff has just been developed within the past 50 years. So it's like explosive. You know, they knew the sky and they knew that stuff, astronomy and all that Tom talked about before, a long time ago. But, you know, now this is just really recent. So this is really transforming the idea of the handiwork of God. It's not just the heavens, it's in the cells too. So the last thing are the city gates. That's what we'll look at. So the city gates, outside of the cell is watery, inside of the cell is watery, and the membrane, cell membrane, has got a lot of molecules just stuck together. It's not contiguous, and what holds it together is each molecule has got a hydrophilic end, meaning it really likes water, and a hydrophobic end, which really hates water. And so the cell wall is held together because these inside molecules do not want to see the water. So it's if it was just all hydrophilic, the molecules would just float all over the place because there's nothing holding it together. This is what holds it together because these will not let go. And so you've got an contiguous cell wall because of the structure of these molecules, which it's interesting that they have those kinds of things, and that's what the cell membrane is made of just by chance. So this is how that's held together, the cell wall. And it regulates what comes in and what goes out of the cell. They have cross-membrane proteins that I'll talk about a little bit in the next slide. And they communicate with other cells. So this is a picture of a blown-up picture cartoon of a cell wall. And you can see there's a lot of cross membranes. Again, everything's guarded. If there's something that comes in and goes out, it has to pass muster for whatever the thing is. It doesn't just leak back and forth. It's very, very highly regulated. And there's one that I think is really cool, this cross-membrane protein, the signaling protein. So what if you are outside of the cell and you want to get a signal to the inside of the cell, but you don't want to go in, you just want to send a signal into the cell. And what happens is there'll be the signaling protein comes over and it would sit on a protein like this, that's a cross-membrane protein, and as it sits on this part of the protein, binding to that protein changes the conformation of this end of the protein. When this conformation changes, that changes the conformation of this side of the protein. And then this side of the protein, when the conformation gets changed, the shape of it gets changed, that sends a signal to the inside of the cell. So it's kind of like a little communication system without actually going in to tell the things. It was just, and I think that one's really cool. So where does that leave us? I think that leaves us with, this is what Darwin knew. This is what he didn't know. And I think he should have known enough just by the heavens and what was going on in the heavens and looking around. But people nowadays who know all of these things, I think they have no excuse for saying, oh, it happened by chance, you know, so. The other thing I was going to tell you is there is a movie. Do we have time? I might have one on my computer. Yes or no? It's about 10 minutes. No?
No, we don't have 10 minutes, we have about 5.
I've got
Okay.
questions too.
Okay, all right. Well, here's the references. I have to put my references up just to be. So you can Google Inner Life of the Cell. Google that, it'll come up. There's a shorter version, and I think I've got the longer version on here. It will show you some of the pictures I grabbed from the movie, but it's a little movie that shows all of the stuff that I just talked about. So it's Inner Life of the Cell. and it's put out by Harvard, I think. Yeah, Harvard. It's a very, very, very well done movie. So, that's it. Questions?
If you ever watch Ian's commercial, it always has words, but wait, there's more. And I think there's things that Betsy just didn't have time to cover. You think about the roads. Well, yes, you need roads, but there has to be a traffic department that lays the roads out as a way to go where it comes from and where it needs to go. The cell walls or the city gates, they have to have controls to do certain things but not do other things. How did the logic, so there's effectively computer programming and planning that's all behind the scenes to make this work, so that the evolutionist who says, oh, it just happened, it had to be this way. Yes, it had to be this way, but there's no way you can go from disorder to order to make this happen, and that's what they're ignoring.
And, well, how does a CRISPR work?
You know, that was there after I retired, and I don't remember. I really don't know. It's some techniques that they use that they can go in and modify certain bases of a cell of a gene, but I don't know the methodology. That was later.
Because it looked like near the beginning of your presentation, you had some of the information removed.
Yeah.
Like that block information, that was taken out. Is that how a CRISPR works then?
I don't think so. I think they actually modify the bases that determine the protein.
Okay.
So it might be. In fact, those introns that are taken out, those are important because what's in those introns is not junk. It is things that are controlling the gene, so that if there is there's called the splice site on the end that the protein knows to cut there and not cut where the exon is. The exon is the one that gives the protein information.
Okay.
But if there's a problem with that splice site in the intron, you could get cancer.
Yeah.
You know, there's everything. If this little thing is wrong, you can get cancer. If that's wrong, you can get cancer. And so there's so many either normal functions and there's the feedback mechanisms that are all going on inside the cell. And if something goes wrong, you get cancer or something else. You get some other genetic disorder.
So, then there was another question I had. Like when a cell's dividing and if it's interrupted by radiation or something when it's dividing, it will deform. Could it turn to a cancer, or you know what I mean?
Yeah, if you
It's at its weakest point.
well, like 3 Mile Island. You know, for example, I've had 3 different types of cancers, and I was within 5 miles of 3 Mile Island, and I thought, well, that should be lung cancer. But no, I was talking to the doctor, and he goes, no, there's Rocky Flats. He sees people who were at Rocky Flats had all these different cancers, and I've had, you know, so it might have been, or I might just, but God's been good because I'm here. And so there's the radiation and if it destroys some of the DNA in a cell, if the cell's working properly, it will signal the apoptosis to kill itself.
Oh, okay.
It's just when those housekeeping genes, or not housekeeping, the monitoring genes,
Yes.
see something wrong. If there's something wrong with those genes, then you have a problem.
Okay, so then somehow the monitoring genes wasn't working properly, and it's not necessarily radiation either then, not necessarily.
It could be,
but
yeah.
Could be a lot of,
Yeah, something was read wrong, something happened, and
Yes.
yeah, there's the fact that we are all fairly healthy and functional says a lot about what goes on in that cell, because there's a lot of systems and mechanisms goes on all the time in each and every cell of our body. Anything?
All right, did you feel like you just finished up with high school or college science class?
2 years of college.
A lot of information, but I think the benefit that comes out of all of this is the reminder that our bodies have such an amazing design, even down to levels that, I mean, we just have absolutely no clue, right? There are so many things going on that we can't even see, and God has orchestrated all of these things. So if you've ever tried to build something, you know how hard it is to get everything to work all at the same time. And in the kind of things that are going on inside of our bodies, they just couldn't have evolved. Millions of years, not enough time. Billions of years, not enough time. Besides, where does the energy come from? Where does the information come from? There's just all these questions that really, when you step back and look, any person who truly considers the implications of all of these things has to come to a single conclusion, which is there has to be an all-wise God who did this, because there's no other way for this to happen. So I know there's probably a lot that went over our heads this morning. If you're like me, I'm like, oh, I remember that word from science class, but I don't remember what it does or where it is or what happens. But
I skipped a bunch.
yeah, for real. But at the same time, we also remember then that there is such a high level of complexity that we don't even fully understand yet. And God is in charge and orchestrating all of it. How wise and how powerful he must be. So Betsy, thank you for that. I think that helps us feel a little bit smaller and helps us see that God is a lot bigger than maybe we knew he was even before we looked at this morning. So we'll go ahead and take a break. We'll come back for worship in about 10 minutes, and we'll have a chance for a little bit of fellowship.